And our next speaker, certainly no stranger to a lot of us in the room, and I've referred to Emil frequently as one of the researchers in PERC that really adds a certain amount of glue to the whole organization. He does interact very widely with a lot of the groups that have made a major contribution, and I think he really has helped us an awful lot in the transdisciplinary nature of this whole program. Emil joined GIFS and the PERC program at its inception, at the inception of PERC. He was previously the Director of Strategic Scientific Development at the Canadian Light Source, and has maintained those strong contacts with the Canadian Light Source to help us collaborate at that level. But he's also taken some very new initiatives in Pillar 2, the what we call the image and data acquisition technologies, involving a number of different types of radiation to help us get images that will be useful in plant phenotyping. And the title of Emil's talk is visualizing plant parts using penetrating radiations.
My business is innovating tools to do plant characterization or plant part characterization with things other than visible radiation. So the first thing about this is the team growth. I see that some people have missed out on that. I did this in the, I guess I did I did the wide format, and so some things might not work on this presentation. But the team that you see here, 10 years ago, or 12 years ago to be exact, it was only me. And then we developed with Jean-Claude Kiefer and Sylvain Formeau at the ALS, the Advanced Laser Light Source in Quebec, and it's grown substantially since then. We have a large contingent from GIFS. My name should be on there as well, and Paul Arneson, Dave Schneider. Anyway, you see that one of the things about GIFS is that, and PERC in specific, is that it has encouraged a very strong interdisciplinarity. And you see that the group of people, there are some recognizable names: Elliot Meyerowitz at Caltech, Mike Dixon at Guelph, Ron Rogie and company at CNL, and so on. We've developed a good research team across the country, all looking at using minimally invasive imaging for plant characterization. Here are the techniques. I thought I would. I'm a physicist, by the way, so I thought I would start by going through the various different techniques and then talk about some of them here. So the first technique, and you've seen some beautiful examples of this, positron emission tomography. That's one of the techniques that can be used for characterizing both structure and function in plants. Nuclear magnetic resonance. I insist on using the word nuclear, even though it's called magnetic resonance imaging now, because it is a nuclear magnetic resonance technique. This is using a high field and imaging materials using the interaction with the magnetic moment of some particles. X-ray absorption, there are two ways of looking at plants with X-ray absorption. One is computed tomography, and the other is looking at the effect of X-ray absorption and correlating some of those effects with structures that you observe. So there's X-ray absorption near edge structure, there's X-ray absorption fine structure, X-ray fluorescence, and X-ray diffraction. I will only talk about X-ray fluorescence and computed tomography in this talk. And then finally, the other two things you can do is neutron scattering and neutron absorption. Neutron scattering is very similar to X-ray absorption. It's a way of doing computed tomography in what is called an extinction image, where you look at what doesn't get through your sample. And then neutron absorption is a way of looking at specific elements by capturing a neutron onto them and detecting the consequence of that capture. This is the general method, and it's not meant to be flippant, but you start with an understanding of the physics on the left, and you get some kind of radiation. You put a plant between the radiation and your camera, and then you follow that up with some data processing, and sometimes followed up with the brain. And the brain is probably the most important part of this topic, but this is the general flow, and it hadn't occurred to me until just recently that I was assuming something that maybe some people don't know about the process, and that is that you do have to have radiation. You do have to have a plant that you right now that you manipulate. You rotate that plant, and then the other technologies are in the system. But this uses a different kind of plant phenotyping. We're looking at plants that are grown in controlled environments. It's been my belief as an arrogant physicist that one of the reasons that it's been difficult to forecast plant growth from the controlled environment to the field is because we haven't had good measurements of the controlled environment before, and I'm hoping to address that problem. So for positron emission, you use a proton. The source of these techniques for positron emission, you need to have a proton cyclotron. For most of the isotopes, you can make some with other techniques. For the short-lived isotopes that are most interesting, the proton cyclotron has to be adjacent to the plant growth facility or the plant analysis facility. For MRI, you have a high magnetic field, and you have to have plant and soil with no or very limited ferromagnetic materials in them to confuse the MRI imaging. For X-ray absorption and scattering, you need to produce relativistic charged particles in free space. And it's important that they not be in a matrix of any sort. They need to be free particles. And then for neutron absorption scattering, you need a neutron source and the moderator to adjust the neutron temperature. You want to bring, for most purposes, you want to bring the neutrons down to thermal temperatures or lower so that they interact most easily with the light elements that are of interest in plants. So positron emission tomography, you've seen that. Positrons and electrons are antiparticles. When the positron is emitted, it travels a mean-free path. It's typically about 1 millimeter, and this is the intrinsic resolution limit for PET-CT. Annihilation releases back-to-back energetic photons. Tracking them tells where the annihilation event occurred, not where the positron was emitted. And this enables both functional imaging and structural imaging. Nuclear magnetic resonance, as discussed, you have a high magnetic field that interacts with the magnetic moment of a particle. It can be used to track any stable particle with a known magnetic moment. I won't talk about these two in this. X-ray absorption, usually use electrons as a light, stable, charged particle that's easy to accelerate to relativistic energies. When relativistic electrons in free space are bent, a photon must be emitted. And if the energy is high enough and the rest mass of the particle is low enough, like with electrons, these photons are often called X-rays. And they were first observed on an IBM synchrotron, and so they're called synchrotron light normally, but they're just X-rays like everything else. I wanted to talk about this a little bit because one of the things that I've brought to this field is the laser Wakefield Accelerator facility. And it has some unique characteristics. It produces the same synchrotron light that you produce at a $500 million facility like the CLS synchrotron, but it costs a couple of orders of magnitude less to do that. So you can put these things into real plant growth facilities, and you can actually realistically contemplate having a plant characterization facility in your greenhouse. Rather than bringing the plants to the facility, you can put it where they grow. Every element has a unique X-ray absorption signature. The absorption directly produces attenuation images and element and chemical state specific maps. And X-ray scattering, which was mentioned briefly the other day, gives structure at the nanometer level. Neutron scattering also provides structure at the nanometer level. Neutrons do not interact with a charge field. They interact only with the massive parts of the fields that they see. Scattering can be used to create attenuation images. That's what I do at the neutron sources. Neutrons can also be captured on nuclei, and the resultant high energy capture gamma can be detected for element-specific mapping. So what does an ideal plant characterization facility look like? I'm 5 years into, or 6 years into designing this, and I really think I know what needs to be done. One is that the plants need to remain vertical. Large vertical range of motion is required, but we really don't want to rotate the plants. You really want to rotate the source around the plant. You don't want to rotate the plants. Plants under optimal growth conditions will grow about 1 micron per hour, and that means that if you're doing a high resolution CT image, you really have to do it quickly. And you really have to do it in a way that the plant is growing naturally so that you can possibly back out these effects if you want to. So you can't lay the plant down, it needs to sit still, and it really does not want to rotate either. The environment where you work should be plant-friendly, well-illuminated, controlled humidity, controlled daylight cycles, and ideally a controlled atmosphere. You want to nurture plants in the environment in which they're imaged. No magnetic materials in this environment to enable simultaneous MRI, neutron, X-ray, PET imaging without moving the plant. This is really ambitious, but a laser Wakefield accelerator facility is an important part, and it allows you to produce both intense tunable X-ray beams and intense tunable or intense adjustable neutron beams, and so you can do both imaging modalities at the same point. I was talking to Rich yesterday, and he has some ideas about simultaneous measurements. I want to assure you, Richard, that these slides were produced for a talk that I gave in France at the beginning of this month, so this is, we're not copying ideas. It's just that this is a different approach here. Some reasons to image the rhizosphere more carefully in natural non-disturbed soil. You guys know this, I didn't, but I've learned it in the last few years. Fungi are moving from the equator at about 8 kilometers per year. Most food crops will be pest saturated this century, and nutrient use efficiency needs to be improved to ameliorate things like algae bloom made worse by over-application of fertilizer. This is one of the reasons why you want to do this in intact, undisturbed soil. And now I'm going to show you, I tried to group this into 2 groups. I'm going to show you some neutron images. Neutrons are highly sensitive to water and to light elements. Neutron capture, which I won't talk about, can also be used to identify specific elements in inhomogeneous structures and in large pots of soil. This is one of the early slides that I borrowed from Jeff Warren and company at Oak Ridge National Lab. This is just to show a capability that exists, and that is that you can actually, while you can't see the fungal hyphae directly, you can see the water that's associated with them, and you can see it associated with roots, and you can do things to turn functions on and off in the soil. The triangular pattern here is just from the way the soil was poured into the device. It's nothing important. What this illustrates is something important, though. You're actually looking through an aluminum box. The aluminum is about 1/8 of an inch thick. The sand is about 1 inch thick in this picture. And neutrons see neither. They see just the water and the light elements which are in the plants and in the roots. And so it's a beautiful system for looking at plants and doing functional studies of plants. There are 2 different plant varieties in this. There is sawgrass, and there's another. I think the big seeds are young corn plants. Oh, I touched the wrong one. Okay. So this is just showing one of the examples where you can do a dynamic study. If you look where the circles are, you'll see that roots are growing into those circles. These were taken a couple of days apart, because this is about 50 micron per pixel, so that's a significant growth that's happened there. This is showing another way that you can look at. You can look at water flow in these rhizoboxes. And you can do dynamic studies where you introduce water and other nutrients, and you can watch roots growing in their direction. This is an example that I did. I apologize for those of you who've seen it before, but I try not to show images that I want to publish. And so this is one that's been shown on, and so in the public domain. I don't care about it anymore. This is a study that I did to try to see if I could separate. These are living plants, and there are 2 wheat seeds and 1 pea seed. The massive amount of roots is from the pea plant, but you can see the different structures. And in this structure, without doing anything, I can tell you, without doing anything to the plants and keeping them living, I can tell you within a few percent of error what the root biomass is. I can identify all 3 root systems, keep them completely separated from each other, and there's no brushing and no watering and no other things done to this. It's just you take the picture and you go. And this is one of the absorption images, and that's a crude picture of the root architecture that's derived from that 3D stack of images. And then this is just showing, this is in canola, and this canola is diseased. You can see that the root is very much thinner in diameter than the stem, and that canola plant is going to die soon. On the other side was a wheat plant. Just going to show you another example. These are rhizoboxes. This is where we're looking only at water flow in soil. And so we deliberately created some columns of different density soil types. There's fine sand on the outside, sand and clay. And then there are a series of about 8 wheat plants growing in this. You don't see them right now. But you can see that the clay hangs on, and we've stopped watering a couple of weeks before this was 3 weeks before this was taken, and you can see the results of that the clay hangs onto water, obviously, and the sand and fine sand don't. What we're trying to see, and it's indicated a little bit here, is whether or not the uptake of water from soil was significantly different depending on the size of the sand granules. And you can observe that down near the bottom right-hand corner, you can see where the roots are and where the soil is drier because of the presence of those roots in that area. And we can quantify this as well. So this is after I've done a water event where I added water at the bottom and tried to look at how it moved. It moves rapidly up and out through the box. And then this is 20 minutes after hydration, and I've subtracted much of the signal. You can see where water is starting to accumulate in the leaves. Leaves are hanging down outside this box, so the structures at the front are wheat leaves that are hanging down. This is 6 hours after hydration, and what you can see clearly is that in the fine sand, the roots have picked up a lot more water a lot more quickly than they did in the coarse sand or in the clay, and we can quantify the water behavior of these different sands, and it's an interesting little experiment. And this is not showing, sorry, I should try to go back if I can. Whoops. Okay. This is showing normal garden soil. This is not sand, and we're just looking at, so this is the same soil throughout the box, and you can see the root structure and how the roots have dried out the soil as they're exporting water up to the plants. This is looking at plants with neutrons in commercial ProMix. This is probably the hardest thing that you can think of doing because there's so much organic junk around this. So there's 3 plants, pea plants in ProMix, and the challenge was to extract the root system with all the organic junk around. And you can see as I work on this, you can now follow the roots all the way down to the bottom, and you can see the root with most of the organic noise removed. And that's what this is looking at plants on the laser in Montreal. These are some poplar plants. I was looking at, and this is actually with neutrons, but it's the same poplar plants. And this is before and after hydration. And the green is heavy water, the blue is light water, but nothing is done to the water other than hydrating the plants. You can see, I guess you probably can't see directly on this, but the stalk has expanded a little bit, and the green are smaller than the blue water because the hydration has displaced normal water and it interacts with neutrons less. Now we want to look at some X-ray photon images. Here we have a standard image that I showed to show the power of laser Wakefield. The image on the left is the same wheat head with a contrast agent taken at the Canadian light source. The image at the right is with a single flash of the laser. The image at the right is considerably more detailed with no contrast information than the best you can get out of the synchrotron. This is a bit of a cheat because the synchrotron beamline is too high in energy, so it doesn't easily image seeds. And I have a broader band energy distribution. But this is a single fire of the laser. This was done some 4 years ago. And you can see the benefit. The beauty of imaging with a laser is it's a much better optical system than you can get at a synchrotron. And this is exemplified here. The important curve is the one in red under the flat field for imaging. That shows the distribution of the flux of photons across the entire field. That field is about a 1 by 1 centimeter square right now. We've adjusted the optics for that. I've done some images at synchrotrons and other places, and because the synchrotron has an extended source, you can't get a beautiful flat field structure like this. So the imaging that one can do with a laser Wakefield, the source is about 1 micron in diameter. You get a much more optically pure and much better image when you use a laser as a source, a laser Wakefield as a source rather than a synchrotron, and that's what you saw in the previous view graph. This just shows the way we measure energies at the laser because we don't have a monochromator. The beamline is as simple as it can be, and we're trying to look at edge effects on the right. We have something we're just imaging a channel, and we're looking at other edge effects. This is looking at an array of nylon wires. The important thing to take away from this is that you can do a trick called phase contrast enhanced imaging. You see the edges of each wire very, very clearly, much more clearly than you see the absorption in the wires. And edges are what's important if you're trying to see the smallest roots in heterogeneous media. So it's a very important ability to be able to do phase contrast enhanced imaging, and we do that very well on the laser in Montreal. This is just an example to show how you don't have to use neutrons. You can use X-rays to individually count all the water channels in this stock, and you can also quantify how much water is in each by simply doing a stressed image and then an image after hydration, and you can align and subtract, and you can work out how much water is in each of these channels. This gives an example. I alluded to phase contrast enhanced imaging. This is the same picture. The top, we've just looked at X-ray absorption. The bottom, I've looked at the phase effect. You can see how much more clearly you can see roots. The biggest thing there is 300 microns, and the smallest that you see clearly is 10 microns. There's a 7 micron beside it that you barely see, but the smallest thing you see is 10 microns in diameter. This is just a Jerusalem artichoke. I just had this because Paul Arneson wanted to see it, and you can see this illustrates one of the issues that you have to do when you're getting an image stack is you have to normalize properly. This is not normalized. This is just raw what comes out of the beamline. Soybean leaf, I wanted to show an example of another technology. You saw some beautiful examples of looking at other things than just absorption. And this is looking at soybean leaf with confocal X-ray microscopy on a beamline that I built in Chicago that is co-operated by the Canadian Light Source. So this is a soybean leaf, and I think you'll see that's just looking at the scattering of X-rays. So, and the piece we're looking at is up here in the corner. And this is just looking at one of the veins in that soybean leaf. We're interested in looking at an element-specific map around the vein in the soybean leaf. And you can see that we have, with this confocal X-ray microscopy, we've identified a whole series of elements and how they're distributed around around that leaf. And what's not shown clearly here, but is something worth noticing, is that this virtual section is virtual. We've created a section without actually slicing anything into the leaf. So these sections that you see are below the surface of the leaf. They're inside the leaf, and we've actually done a vertical section, a vertical slice through the vein that you see without cutting the leaf. This is all done digitally by just moving our confocal probe. So we're moving the confocal probe in a vertical scan through the leaf. And this is one of the early things that we did. There's a 7-day-old canola plant. There's an X-ray image at BMIT, and there is the reconstructed same plant reconstructed after a series of X-ray exposures. And you can see that instead of just one plant in there, there's actually 3 seeds. One is erupting, one is just starting to erupt, and the other is grown. This is just an old, this is the very first thing that I did in imaging plants at the CLS. Phase contrast. This is another illustration of the beauty of phase contrast. You can easily count all of the vasculature in that in the stem there with phase contrast, and not so easily with absorption. And then you can see that the phase contrast it gives you a more than 10 times better sensitivity to edges within the plant. And this is a phase image of a pea seed. Again, this is a single shot done in Montreal 5 years ago now. You can see that you can you can identify structures across the surface, across the seed. And this is doing the same thing, phase imaging of canola roots. And we talked about the wheat fusarium. I'm trying to speed up because we're trying to get back on schedule here. This is looking at the wheat fusarium with phase. So the previous stuff that I showed was just looking at the X-ray absorption. This is looking at the phase, and you see that we can elucidate all the edges of all the structures inside these seeds much more clearly and beautifully with looking at phase information. And then this is another example of looking at water flow through this drought-tolerant poplar. These were provided to me through AAFC. Raju Sulanaya Kanahali is the guy who grew these for us. And you can see that you can count each individual. On the right-hand side, you can count each individual water channel, and you can follow easily by looking at subtraction. We can quantify how much water is flowing through each of those channels. This is a final study. This is done for the guys at Caltech. This is looking at a single-celled algae. The single-celled organism is big enough that you can wrap it around your waist if you want, even my waist, and I'm not thin. So this is a single cell, and they're not supposed to. The issue is how cell walls grow when how cell walls grow when they're when they're a single cell, and there is how structures grow inside this. Nothing is known about this. This is the first time this algae has been characterized. This is just a visible light picture of a region that we're looking at, a 4 by 4 millimeter region of this algae. And this is showing the different elements that we identified and how they're differently localized within this single cell structure. These are parts of the algae that function as roots and stems, and you can see that the distribution of this set of minerals or elements is significantly different. Prior to this, there was no evidence that vascularization actually occurred in these single-celled organisms. It's clear that it does now, so we're working to extend this a little with some more detailed imaging. This was done at Cornell. These are just a bigger picture of each of the individual elements, and and then this is the last thing I wanted to show. This shows you how well we understand these materials, how we can make these elements. This is looking at a different portion of the caulerpa. This is a stem of the caulerpa, and what you see here, the different colors are different modeled peak concentrations. These are the data is behind this, and there are no free parameters in this calculation. So we actually exactly fit the X-ray fluorescence spectra of this algae. So we really do understand what's happening and where the different elements are. And this is an example of what we can do. I tried to show what we can do with both X-rays and neutrons and why it's important. The laser Wakefield facility is uniquely capable of generating very intense X-ray beams that can be rotated around the plant. So you don't have to rotate the plant anymore. You can rotate the X-ray around it, and that, I think, is a very important capability to have in our multi-modality imaging station that I hope to eventually see here.